With that, again, a formal welcome to the second SentroConnect live webinar. Today's webinar is focused on a specific PAT application, here the use of the NIR in the tablet press. And we have two presentations, one to focus on the technical and the other on business considerations on the implementation of PAT in the tablet press feed frame. With that, I would ask the two presenters today to briefly introduce themselves. In that case, Owen, I would ask you, can you give a brief introduction about yourself?
Sure, Hubertus. Hi, I'm Owen Rehrauer. I'm the president of Sentronic US Corp. I've been with the company since its inception in 2022. Prior to that, I was a Sentronic global partner since about 2019.
Thank you, Owen. Second presenter today will be Uwe. Uwe, if you want to share a few words about yourself.
Yeah, hello everyone. I'm Uwe, co-founder of Sentronic and with the company now for 32 years with a background of electrical engineering. Yeah, I'm happy to participate this time.
Perfect. Thank you, Uwe. My name is Uwe Rehrauer. We see two more persons joined, at least on the panel, which is Sebastian and Adam. Both will be familiar for those of you who have worked and known Sentronic for a while. They will be happy to support us at the question and answer session, which will be at the end of today. However, we will skip a brief introduction to get straight to the topic. The first topic today will be on the The first topic of today will be the technical considerations for the implementation. No, see, now I'm confused myself. The first presentation today will be on the business implications for the application of PAT in the tablet press feed frame. This is going to be presented by Owen, who has prepared that topic. With that, I would ask you, Owen, to go ahead, share your screen, and we are looking forward to your input. For those of you all attending, I would like to mention that questions will be answered at the end. So please, if you have any questions, don't hold them back. You will find in the meeting tool, in the webinar tool, a Q&A session, and we are looking forward to already get your questions. We'll collect them and we'll address them at the end of today. With that, Owen, you can get started.
Thanks, Hubertus. Can everyone see my screen?
Yes.
Fantastic. Thanks for the introduction and thank you for everyone who is attending. Today's discussion is going to be around the business case for NIR PAT in a tablet test feed frame. This is a multifaceted topic that would really take me quite a bit more time than we have available today to cover in a fulsome way. So I'm going to cover this at a very high level, although I will provide links to academic or industry articles that deal with this topic in more detail for the various areas we're going to discuss. In brief, we're going to be discussing what value PAT can add as a business case for enhancing manufacturing. We'll also discuss some of the cost and risk associated with implementing it and ways to evaluate whether or not using NIR PAT in a tablet test feed frame makes sense for your application. We'll go through some example business case studies where this has been done successfully, and then we'll have some time for some questions. Let's talk high level about what value PAT can add to a manufacturing organization. Well, we often, as PAT SMEs, will discuss technical advantages, technical capabilities, the ability to measure things like blend uniformity or content uniformity. I think it's worth taking a step back and talking about what benefits those provide to a business. These will be familiar to many of you, but these benefits can include reduce the overall cost of QC analysis, reducing inventory of product needed by an organization, improvement on yield, an enhanced process understanding, and some potential filing advantages, at least in the USA. By that, I mean if PAT enables you to declare your manufacturing operation advanced manufacturing, you can get some expedition in your filing for, again, a US filing. I do want to call special attention to the inventory savings, as this can be a non-obvious place where PAT can have a big advantage for an organization. I've listed a literature reference here where several brand name pharmaceutical companies have reported in 2009, 2010, billions of dollars in savings. I have a friend who is a high-level person at a brand name pharmaceutical company who likes to say, if you want to make the case for PAT, talk to your supply chain. You might find that PAT can have a larger impact there than you might see just from the analytical cost alone. I think that's worth keeping in mind when assessing the potential benefits or downsides of PAT. These are all the wonderful things PAT can do, though, although we should be clear-eyed when we discuss the potential cost and risk of PAT. There's some obvious cases here in general for PAT. There's a cost associated with the hardware, there's operational cost. PAT often requires specialized labor. If you need to calibrate or maintain a chemometric model, there's some material and labor cost associated with that. PAT can have maintenance cost itself. In addition, it also requires specialized data management to get PAT data out. Oftentimes, material or data management systems for a manufacturing floor are not designed to take in spectroscopic data, and we've seen several specialized solutions to management. This also has a cost, though. And last but not least, organizational comfort with a PAT approach. The pharmaceutical industry is a fairly conservative industry. We've been doing chromatography since the 1970s, and that's been a consistent way to get the manufacturing process filed with regulators. There are many business advantages of PAT. However, there can be some risk associated with that, particularly in terms of filing. Additionally, while there are certainly centers of excellence in Europe and America where there are lots of people who have special experience with PAT, we do have to acknowledge that in some areas of America, some parts of Europe, and the rest of the world, there may be some challenges getting specialized labor that can perform the sorts of work we need to be successful to implement PAT. There can be questions, particularly with some emerging PAT developers, on the overall business stability of the instrument provider. With Sentronic, we've been in business for over 30 years. We don't tend to get that question anymore, although Uwe can tell you in the early days of Sentronic, that was a question, and an understandable one. Last but not least, we should also keep in mind the impact of using PAT on the overall operational flexibility of the manufacturing process. If strategically it would make sense to hand a process over to a CMO, can a CMO implement a PAT strategy you have? It may be possible to enable that, but that may come with some additional costs, which we should keep in mind when we're trying to optimize our return on investment or overall business efficiency. That's a very high-level discussion of PAT, but today we're interested more in discussing what we can do in an NIR feed frame. So what are the application cases for a PAT measurement in a feed frame, and what are the business advantages of using these methods? For starters, we can measure blend uniformity as an in-process control when we use NIR PAT in a feed frame, which allows us to catch deviations as they happen, potentially allow for them to be corrected, which can save a large amount of material or potentially save a batch or a continuous run. Sentronic has quite a bit of experience using this for continuous manufacturing or helping customers deploy this, excuse me, as part of a continuous manufacturing process. And we're seeing some emerging interest in also bringing this technique to batch manufacturing, which is fantastic to hear. Making measurements here can also enable an RTRT approach, particularly for content uniformity or dissolution. And I will say enable here rather than allow you to make it from the NIR alone, because oftentimes, almost always, the NIR data needs to be supplemented by other process information. In the case of content uniformity, this might be tablet weight, so we can assess the overall milligrams per unit dose provided to a patient, the variance there. And for dissolution, we may need additional information like tablet thickness or hardness. However, if we want to realize the benefits of the maximal reduction in inventory, move material through our process faster, this can be a big business add, although, as we'll discuss later, can also come with some larger costs and potential other ways we might implement this approach. We previously mentioned the potential filing benefits, and I should also mention this provides a tremendous amount of process information for us, which we can use to help ease process transfer from one site to another. These are all great, but we should also keep in mind the potential cost and risk when we're deciding whether or not we want to implement this method. NIR, as we previously mentioned, can include some non-trivial costs, particularly for a novel molecule. We may not have economized the actual manufacture of the drug substance. So as we're making blends of drug product, they can be quite expensive from a dollar per kilogram perspective. There can also be challenges with equipment availability. If we're talking about making measurements in the feed frame of a tablet press, is that tablet press always going to be available for us to do the method development we need to ensure the successful deployment of PAT? There are strategies one can employ to avoid this problem, namely the use of process simulators, but that is an additional cost we should keep in mind when we're assessing the overall cost and benefits. If we decide to use PAT in a method that would be classified as high criticality from an ICH perspective, there also may be some risk to the manufacturing uptime. To put this plainly, by this I mean if we use PAT in a way that we are making product quality decisions and that PAT goes down for whatever reason, there's an instrumental deviation or there's a model deviation, we may not be able to manufacture until that PAT is ready for use again. We should keep this in mind as we evaluate all this. There's lots of ways we can approach this problem, though, and I would say there is no one-size-fits-all solution. Having said that, we see three broad strategies of deploying NIR PAT in the feed frame, and I'd like to talk through the pros and cons of each of these strategies. I will say at the start, none of these strategies are something that you will implement and will never change or are immutable. Oftentimes, we see customers start with one strategy, for example, a very simple guardrail PAT approach, get some organizational comfort, progress on to an IPC, evaluate the potential pros and cons, and if it makes sense from a business perspective, go on to implement RT-RT. At a very high level, I would say it's important to consider the overall strategy of PAT deployment rather than just trying to deploy the PAT for a one-off situation and then not think about it again. So what do these strategies entail? One approach I will call guardrail PAT is something that I'm seeing customers who are interested in PAT but have a low organizational comfort with the method use to evaluate and get a feel for whether or not PAT is right for their business case. Oftentimes, this involves making PAT measurements for information only, so not making formal process decisions using the PAT, but using the PAT to catch potential large deviations from assumptions of manufacturing. For example, in a bin blending process, this might be making NIR measurements and using it to validate that all ingredients of a blend were put into the bin blending operation. If you can catch that and save a batch, the cost of that batch alone might make return on investment for the PAT. To describe what the graph on the left of the screen is trying to describe here, this is not from a real business case. This is going to be qualitative for the three strategies. This is a low cost in terms of dollar value relative to some of the other ways we might implement PAT, but the benefits have a lower ceiling than some of the other benefits we might realize for the business with other strategies. As we mentioned before, though, this might be a great way to start a PAT journey for an organization, particularly one that maybe is skeptical from previous deployments that did not hit return on investment. We also see customers using NIR PAT in the feed frame as an IPC. This has a greater development cost because we typically are calibrating chemometric models and using them to make process decisions. Those models need to be validated, and all of that comes with non-trivial cost. This also tends to be something that ends up in a filing. This can be an advantage. Getting additional time on a filing due to expedition can have a very large dollar value, particularly for branded manufacturing. We have seen this very often in continuous manufacturing as customers feel that they must include NIR PAT, and regulators encourage them pretty strongly, at least in the Americas and Europe, if they are using a continuous manufacturing approach. This can have an advantage depending on how this is deployed in terms of savings relative to laboratory methods or to the aforementioned patent savings. I will say we are seeing some emerging approaches here in how this is deployed to minimize the cost of the NIR method. Namely, we've seen publications from places like Pfizer, I put the reference on the screen here, where a fusion of NIR PAT data as well as soft sensor data is used to reduce the overall calibration burden of the NIR PAT method, and by that reducing costs, reducing time it takes to develop, to maintain an in-process control at a more cost-effective way. I would recommend, if you're interested in that, reading the attached paper to get a better idea of what the possibilities are, particularly in an advanced manufacturing, i.e., a situation where you have lots of other soft sensors over your development floor, to take a look at this. Last but not least, we do have several customers, several manufacturers in the environment using NIR PAT to enable a real-time release test approach. This has the greatest possible potential to realize ROI, although it does have larger upfront costs in terms of method development. We are seeing some companies start with an IPC approach, evaluating whether or not RTRT would be successful, or what the potential risk of using this method are, and then turning the key to enable it if it makes business sense. So how might one assess return on investment? I debated whether or not to include slides covering the math of net present value and internal rate of return. However, looking at the attendee list, it looked like we were not talking to a lot of business people. We tend to have more PAT SMEs. I will include some references here where the academic way of doing this is covered. And I would implore you, if you're interested in this, to take a look at those and see if there's some advantages. I'll cover those in a bit more detail in the following slides. In addition to what's covered in these references, though, I would keep in mind as you're evaluating this, whether or not you're going to be using a continuous manufacturing or batch approach. Oftentimes that's not included in these ROI calculations. What the regulatory readiness for a PAT filing, particularly if you're a potential manufacturer outside of the US or the EU. I know that not every region has regulators that are used to seeing as many PAT filings, so it may be worth having a discussion. Hearing from some customers, sometimes the regulators are quite excited for the potential of this. You may get some advantages there as well. Also keep in mind future use, readiness of CMOs for operational flexibility. Have some discussions internally to make sure that if you do go with the PAT approach, it's aligned with your overall organization's value and strategy for manufacturing. So talking through the case studies that we referenced here, there is a master's thesis out of the University of Lisbon, which did a great job demonstrating, at least for one case, the value of PAT, which was found to have a positive ROI. This was not a NIR PAT in the feed frame, but the overall process of assessing ROI, I think might be useful if you're interested in taking a look at this for your organization. UPRM out of the Romanok group also has a, in my opinion, very useful business case template for identifying key benefits and risk of PAT. Based upon a literature review, UPRM has been a center of excellence for PAT for some time. And if you're interested in this, I think this paper is worth a look as well. Last but not least, we found some German guy on the internet. Kidding. Dr. Rehbaum had a really wonderful case study in Technopharm. This was a case of using bin blending PAT. And what I think is useful here is it discusses some of the real cost, real risk, real complications of implementing PAT in a manufacturing floor, particularly around data management and orchestration of process materials. So if you're interested in this, I think this is a good sensible look at the potential benefits, but also real-world considerations needed for success of a PAT manufacturing approach. There is much more that I could say, although at this time, I'm going to hand the presentation over to Uwe Kirschner to talk a bit more about the technical considerations of NIR PAT in the feed frame. I'll be happy to take any questions on the business case. I should also say that we are always happy to talk to our customers or potential customers in person at conferences like the IFPAC conference coming up in March of 2026. I should also mention that we'll have a great opportunity to have these discussions at our Sentronic PAT workshop in the US, September 14th to 16th, 2026 in Lambertville, New Jersey. You should be hearing more about that from us here, but I did want to make the announcement as we just booked that. With that, I will hand it back over to Hubertus.
Thank you, Owen. We should go back to rehearsing the presentation so you cannot surprise me with a slide with my face on it. That's a bit, I wasn't expecting that. But it's good. Thank you for the presentation. I think it's a good start. You actually paved the way from the business perspective for the use of PAT in the tablet press feed frame. And the logical extension now is, of course, to take a look at the technical considerations. Before I pass on to Uwe, I'd like to remind everyone that you can already share your questions via the Q&A feature in the webinar system. I already see, and we have a few questions already coming in. So please don't hold back. Put all your questions in. That will make it easier for us in the Q&A session later on to address them, if possible, all of them. With that, I'll pass on to you, Uwe.
Thank you, Hubertus, for the introduction. And I guess you can see my screen?
Yes.
Sure. Okay. Then, yeah, let's start. After Owen looking at the business side of it, I'm trying to give a summary on the technical side of the implementation of NIR PAT in the feed frame of the tablet press. Very briefly, quick overview about the alternatives, then some short history, how this progressed over the time, then considering the mechanical integration aspects of the integration, some little IT and software integration aspects, not that much, and also not very much about chemometrics and calibration. And in the end, I'll introduce some results with the new SentroPAT Compact and compared it with the SentroPAT FO for people who haven't seen that yet, as a potential alternative also for the use in the feed frame. So let's start. This is a schematic of a continuous direct compression line, as it's been used in continuous manufacturing applications since the early days when the pharma industry started with that. Years around 2010 and 11 were the first lines built and being started with. And there are, of course, different positions where the NIR could be located. One is directly at the outlet of the blender step itself or inside the blender, depending on its configuration. This is a very early position in the process, and monitoring the process unit or the unit operation itself. Then other options in the transfer sections where there would be also the option to even integrate a diverter before the tablet press. This also has pros and cons, and today there are solutions for that using, for example, spider wheel type sample interfaces. And then last not least, the tablet press itself, which has a really good sample presentation, and it's also right before compression. So it's the last step before the tablet is being pressed. So it's a very relevant position. So yeah, this all started. I think Pfizer was really driving that development. At least we have no other proofs like that, and Pfizer must have been working on that application as early as 2010, with the first publications coming out in 2013. And around the same time, we had been involved in some of the development ourselves. For example, this is a picture of an RCPE application node done at about the same time using an early SentroPAT FO system in a laboratory type tablet press. And then slowly this developed, and in about 2017, the Pfizer PCMN lines were in preparation, and we had been asked to improve our systems and calibration and instrument matching capabilities. And so we did a workshop and measurements together with the team of the Pfizer colleagues about 8 years ago. And in the result of that, first implementations were made. And yes, Pfizer kept publishing about that. And I can only recommend to really take a look at various application notes or publications which had been following in the years like 2021, 22, 23. They are covering topics like the method transfer between instruments, the capabilities of process understanding from implementing PAT in the feed frame, as well as using it as an in-process control. Not going into details like here. Then, yes, where to place an NIR in the tablet press. And it obviously looks like the best position is from the top in the feed frame itself, as you can see in the schematic. And here the pedals of the wheel are just about very close to the probe tip, and so usually there is a good sample presentation up front. In the early installations, usually micrometer screws were used to adjust the position of the probe to play with the height of the installation. It is questionable if it's still necessary. We think that once the process is understood, it may not be that relevant anymore to keep that flexibility. And yeah, also the question is, are these cutoffs in the wheel really always necessary? We think maybe they help in sample presentation, but I'll show you later that there are also other means to improve data quality, even in this situation without notches. Then there are installations in the field with different types of presses. So I'm just showing you examples here from a Porsche implementation, and alternatively with fette presses, which also allow the height adjustment of the probe itself. And some newer developments also are being made with GA for the next-gen press, where we first time added an extra probe sleeve for high containment applications, where the probe should be able to be taken off the system without opening the containment of the process. Also very helpful for high zip clean in place requirements. So, and the idea is that the probe can just be retracted without opening the interface to the probe. The additional Sapphire window has some loss in the signal, but the amount of it is still acceptable for typical applications. Very important for all the development of methods and PAT evaluations is the use of process simulators like the Expo feed frame simulator. And here, for example, I can show you some technical details of our instrument interfaces with the system. And so I show you the startup process of the feed frame simulator when we were filling up the one simulator with powder for the start of a test run, actually. And you have noticed that the filling happened in this moment. And on the slide, which was just taken at the same time with the video, we see the wavelength monitor of our NIR data at a baseline wavelength, like 1300 nanometers. So in the beginning, high baseline just means we're looking into the empty chamber. And then the filling happens, and then we see how the signal, the baseline is going up and down over time cyclically. So you have seen that even behind the notches, there is some time where there is no sample presentation. So, and to avoid, say, very high fluctuations in the baseline out of this process variation, we implement filters in our system to only use the data which have a really good sample presentation for further processing. And the orange lines just mark the the borders of an individual measurement, which is then saved in the instrument. This is about 1 second intervals per orange here. Taking a look at the spectra, we can clearly see the difference between the process operating without outlier rejection and with outlier rejection. So as soon as the outlier rejection is inactive, the baseline variation increases a lot. You have seen the variance just in the sequence before. And with the outlier rejection tool, the baseline of the same data set is going to be much slower, much more consistent and stable, and also has a lower baseline. So applying the outlier rejection here helps to provide NIR data which have a lower need for spectral pretreatment for further processing. Let's come to some chemometric techniques. This actually is a slide from Adam's part of the SentroConnect webinar 1, where he gave an introduction to chemometric methods in more detail. So I just refer to that. Just with the note that in our current setup here, the really pure statistical methods like MBSD and moving block are not that relevant for the continuous process like the tablet press feed frame. They are more used to monitor process changing or process moving towards an endpoint. So yeah, but besides that, our systems provide capabilities of PCA, PLS, and also lean chemometrics algorithms like IoT to give access to, say, converting the data of the spectra into results. Let's take a look at new developments upcoming. You see here the maybe more known CentroProbe, the RLS on the left side, and the new SentroPAT Compact on the right side. The SentroPAT Compact is a complete system, including the spectrometer, the wavelength reference, the internal reference, a computer, and it's just connected to the network with a power over Ethernet connection. So this means that integrating this into a system is taking away the complexity of handling fiber optic cables and fiber optic connections, which sometimes, of course, adds complexity to a system. So that's why let's look deeper into that. One of the advantages we see for future is enabling a more seamless integration of the systems into industrial and production networks. OPC UA is today a very accepted and commonly used interface, and in the pharmaceutical industry widely deployed. Our current OPC interface is still using OPC DA and uses a commercial OPC UA converter for the current situation. And we are in the development of a native OPC UA server, which is going to be finished within 2026. And this will then allow to run the OPC server directly from any of our instruments, including the SentroPAT Compact. Let's give an overview about the technical comparison between the two instruments. These tests were being executed with the feed frame simulator as well to really do this evaluation under very practically relevant conditions. We were using a caffeine formulation, which was actually blended in the simulator itself. And we were repeating the trials a few times. So we were running both instruments at the same time to always have data from the same process of both instruments types. Here's some raw spectra of both instruments for the Avicel, the caffeine, and the lactose. For the SentroPAT FO as well as for the compact, you will notice that the SentroPAT compact spectral range is limited to 1900 nanometer, which is still covering all the very relevant CH band regions, but misses out the 1900 nanometer OH region and maybe other information. The technical compromise is due to the fact that the instrument cannot cool down the detector like on a SentroPad FO, where there is much more space in the instrument, but actually the detector is going to be heated up to be stable. But this requires that the size constraints of the instrument will not allow to use a cooled version here. Taking a look at the pure PCA data from the compact, this is just steps from 0 to 5% caffeine, and we repeated that trials 6 times in total. You can see in the end we were running out of time, so we were just keeping the stages half the time of before. I think it was 20 minutes before per stage, and then it was only 10 minutes per stage. As time was running out, it was before our PAT meeting in Dresden last year. We also did PLS evaluation of the data. Here is a common method, a common PLS model based on the data of the combined instruments. So it's using the data of the FO as well as of the compact system, just showing both instruments, and there are 5 runs in this case, not all the 6 in the slide here. And the goal of this was also to look at the errors, prediction errors of both instruments under this realistic or close to realistic conditions. And what we have seen here is that for a moving sample, the SentroPad Compact has a prediction noise of 0.06% caffeine in the mixture, while the SentroPad FO had a still better 0.04% accuracy. The difference between both was also about the same for a static sample. Static sample means that the variation coming from the sample itself, because the sample is moving and it's not totally homogeneous, as we usually need to be aware of. But the difference is about the same. So this difference between both instruments is more or less a result of the noise. But when you compare just by instrument noise, the difference looked greater. So it looked like the SentroPad Compact had a 6 times higher noise compared to the SentroPad FO. But looking at it under the lens of a PLS method, which really has a lot of noise reduction internally, that difference is balancing. Yes, with that, I already want to conclude the technical side of my presentation, and I just want you to be aware that we call our webinar series SentroConnect, and the webinar itself is not that much a connection, but we are offering a follow-up session in Teams for everyone who wants to join to that. It's going to be taking place exactly in 1 week from now. And we, the presenters and members of the team, will be there. And any one of you who wants to join to that can do that and ask maybe more specific questions if the Q&A session today is maybe not going deep enough in that. Also, if you're interested, follow us on LinkedIn. Owen, Sebastian, sometimes myself are trying to be active there, and as a company as well. We started this year also a newsletter series, which we call SentroPulse, currently mainly sent out through LinkedIn, in the future also through the website or through an email provider. And last not least, we have started late summer this year to collect our, say, presentations and webinars on our YouTube channel. So feel free to watch for more details there if you are having questions or want more inspiration. Yeah, and with that being said, before it's falling off at the end of the Q&A session, we already want to wish everyone a happy holidays and a good Christmas season. And yeah, with that, I'm finishing my presentation. Thank you.
Thank you, Uwe. Very well. With that, I think we had 2 very good presentations, one on the technical perspective and one on the business perspective. of the application of PAT here in the tablet press feed frame. It is interesting, as mentioned already, it's a nice continuation. We started with the first SentroConnect Live, which, as mentioned by Uwe, you can also review on the YouTube channel. So if you hadn't the chance to join on the first event, visit the YouTube channel of Sentronic and you'll be able to review the recordings. But I think the first question here does fit here. We do have one question asking, what is the benefit of using NIR in the feed frame versus in the mixer, or let's say slash the bin blending stage, reminding here that the first SentroConnect Live was on bin blending. Owen, I can see you unmuted if you want to start off with that.
Yeah, there's some different considerations here. In a batch manufacturing process where you might be using NIR PAT on the bin blending step and then proceeding once that is ready to go to the tablet press, there's a discontinuity between the measurements done on the blending step and the tableting press. And oftentimes there's a non-trivial time difference between those steps. Obviously, it's going to be a bit formulation dependent, but there are opportunities there for things like segregation to occur. So in that situation, I think there's still a clear technical advantage, particularly in formulations that might be prone to it, to measuring in the feed frame of the tablet press. You also, in principle, are measuring more massive material. You're getting a deeper look into your process, whether that's statistically necessary, of business value. It's going to depend on a given operation. In something like a continuous measurement, as Uwe's slides early showed, there are other places one might make measurements in a process. At the outlet of a continuous blender, for example, rather than in the feed frame of a continuous blender. There's pros and cons to each of those strategies. As Uwe mentioned, it might be a bit easier to do things like segregation of material that's non-conforming. The earlier you make the measurement, as opposed to trying to do segregation of tablets as they're coming out of the tablet press. There's just a bit more time there from a mechanics perspective. There's challenges making measurements out of a moving blender. You need to tend to sample the material in a very deliberate way. That can have some added cost and added complexity to your overall manufacturing operation. What's nice about the feed frame of a tablet press is you can put the probe there and you don't need additional hardware. You don't need to make modifications beyond something like modifying the notches of your tablet press potentially. That's a bit simpler, and sometimes simplicity is worth quite a bit just from a manufacturing reliability perspective. So, pros and cons. I always dislike giving an answer that it depends, but in this situation, I think the answer honestly is it depends. But I hope that's an acceptable high level overview.
Thank you, Owen. Just to address the general question in the batch manufacturing, would you then agree that in batch blending, it might be more attractive to do the NIR measuring in the tablet press versus in the bin blending? Or do you say, nah, it would actually be more interesting to have both, that in the bin blending, you determine the endpoint potentially, whereas in the tablet press, you can confirm that the product is like this? How would you argue there?
Oh, that's a tricky one. I think it depends on the individual case, and I think it also depends a bit on if we're talking about branded pharmaceuticals, as in a novel pharmaceutical coming to market under patent protection versus something like a generic pharmaceutical, and what the potential business benefits to both of those situations are. For example, in a generic, it might make sense just from an operational efficiency standpoint to use the blend uniformity for bin blending operations just because of the advantages we discussed in Central Pulse 1, namely the relative reliability of NIR-PAT relative to thief sampling and the faster throughput reduction of that internal inventory to ensure consistent supply. If we can realize RTR on the other side, that can have some real efficiencies there too. I don't want to generalize. I know there are customers that feel very differently about this, but I would say from an economic perspective, I think the case for RTR is generally a bit stronger on the generic side than the branded side. But it's going to depend. So unfortunately, again, it depends answer, but I hope that's helpful in the thinking.
All right. Thank you, Owen. Just for everyone, Owen just name dropped again SentroPulse. Brief reminder, this is the newsletter. So you might want to subscribe to that to also get such insights as Owen just shared. I would like to address a more technical question again on the measurement that probably goes towards Uwe about the penetration depth in the measurement. Can you share some insights or experience, Uwe, on the penetration of the NIR measurements in the feed frame, considering also the moving of the powder bed in there?
Yes, as we have seen in the slide with the absorbance over wavelength or at the 1300 nanometer, there is of course a high fluctuation. Biggest part of that is the distance of the powder bed to the probe surface, so to say, because it's not like fluffy, getting fluffy or a little less fluffy. It's more like a bed moving, at least in the situation we have been facing in the feed frame simulator. It might be a bit dependent on the pedal speed, etc. Besides that, there is density variation, for example, due to the compression of the material in front of the pedals. And after the pedal, there will be either no material or loose material. So using the outlier, we can more or less focus on the higher density part of the data, which is then closer to bulk density usually. So I wouldn't expect the powder to be very different from bulk density. And then penetration depth has been investigated by several PAT experts and practitioners over the years and has been usually calculated and measured in a range of, say, between 0.5, 0.3 to 1 millimeter. Depends on the formulation and very much depends on the wavelength as well, because of the absorption coefficient being very much wavelength dependent. So there is never a fixed answer like the penetration depth is exactly this and that. It's dependent on many parameters. So an estimate is always based on some assumptions and some good, say, rationale behind.
Right. So now if the measurement is taken in the feed frame, we can use the NIR spectra to predict multiple attributes. We already mentioned here blend uniformity, probably also API. There was a question around dissolution. Is there also the application towards dissolution assessment?
Yeah, NIR PAT blend potency measurements can be useful in estimating either a given Q20, Q30, or estimating the overall blend curve, or sorry, blend curve, apologies, the overall dissolution curve. It's still a bit early here. What I would say is you can find mathematical forms that best describe the rate of dissolution of the drug product. For example, you might be looking at something like a Noyes-Whitney description, and you can find process parameters that fit some of the constants in that form. Your plateau is going to be governed by how much mass of material, how much maximum material can be dissolved. NIR can be very useful in estimating where that plateau is going to be relative to other plateaus. That is one piece of the puzzle. There are going to be other pieces there too, and they are going to be very, very, very formulation dependent. So one formulation might be driven entirely by tablet hardness. Sometimes particle size may be important here. Amount of disintegrant, amount of lubricant. It depends. We have seen some examples of people publicly presenting that. I don't want to speak to that. I will let you look at the literature on RTR Disso, and I think you can find many examples.
Right. And if we go back to the API and concentration of API, we again have here a question that asks among the low concentration APIs and identification in IR. I think this is always a challenging topic. We know that it's not necessarily the easiest if you go 2% and below. Do you have any knowledge maybe or experience on the application of NIR in the feed frame, if this is more for high dosage products or across the whole bandwidth? So anyone from the Sentronic team?
Yeah, I would say my rubric when I'm starting a discussion with someone interested in NIR PAT is a formulation 10% or above API is a very, very, very high likelihood that we're going to be able to make measurements there and have sensitivity to those degrees of potency changes. over the way that we typically would want to have sensitivity there. So building something like an 80% target to 120% target calibration model, that generally is much more likely to be successful. Below 10%, getting into 5 to 2%, the answer is honestly going to be it depends. The reason it depends on how NIR sensitive the API is, and it depends on how much the API NIR signature looks like other constituents of the blend. So if you've got an active pharmaceutical ingredient whose spectra of the pure API looks just like the spectra of mannitol, that's going to be challenging. In uh, pharmaceutical NIR PAT, it's rare that we completely miss out from a selectivity perspective. So I haven't had anyone come to me in pharma and ask for a sodium chloride method, which would not be NIR active at all. Um, some of the biologics we've seen can be a bit more challenging. So I would keep an eye there. I'm seeing more in the 2 to 5% be successful, but I will say it is definitely challenging.
All right. Thank you, Owen. Now we have identified a few attributes that are commonly identified or retrieved from NIR extracted. I'd like to come back to a question towards the technical parts, probably again towards Uwe. You did show a few examples for tablet presses. You did show 3 vendors. What is the typical existing automation integration level? So you mentioned that you have the new OPC UA server upcoming, but is currently, do some of the machine vendors for tablet presses already provide an ability to interface also with your equipment or with the NIR unit? And then how likely is it to use that, for instance, for an automatic rejection of tablets based on NIR readings? Can you share some experience and the state of development there?
Yes, we have been involved in a couple of developments integrating the PAT equipment into PAT software solutions like SciPat or SynteQ or PharmaMV. So it's not necessary at the moment, or at least at the current integration level, that there is a direct integration with the press. Very often the NIR is integrated with a PAT management system, which is then part of an overall solution, which integrates the tablet press and all the other equipment as well. The development of the OPC UA interface will just make it easier for tablet press makers and other instrument or production equipment manufacturers to integrate PAT systems like this into their own visualization, so into their own UI system and everything, and it will be more smooth and easier to integrate. I'm not sure if this answers all the question right now. So diverter applications have been developed also, even back in the very early examples where we have been involved. Diverter was part of the schematic, and this has been repeated in other cases as well. So rejecting tablets based on the NIR result is an option which has been implemented already technically, not necessarily directly from our system to the tablet press, but over the PAT system to the tablet press in this case. So I think
one challenge in that is the time you need to process and the speed of the tablet on the tablet press on the other hand. I mean, just looking at the question again, I think it's worthwhile to mention that, of course, the acquisition process, what is the typical acquisition times that you have for a single scan that you get back in a tablet press? What is the common settings?
1 second is a good option. We have users or customers using maybe 2 to 3 seconds and 4 seconds. It is also related to the amount of material and to the volume measured per measurement, because depending on the speed of the pedals, we have a powder movement. We didn't discuss this in the presentation right now, but usually you try to match the measurement time to be at a relevant level of material you measure for uniformity. So unit dose, for example, maybe limit you to a certain number of averages or to a certain measurement time. When it needs to be fast, it can be very fast. So the earliest implementation we did have a measurement repeat of 0.5 seconds to be able to run a diverter behind it. And there, the implementation to the diverter was more or less straightforward from the instrument. So the instrument itself was predicting, and the predictor was controlled by the instrument itself. So long network delays or lag times might create problems here. I think a usual Ethernet connection today isn't that slow. It would probably be fast enough, maybe using TCP/IP or even UDP. But it is possible to go very close to the prediction usually is not that the computing power required for a prediction is not that much for a simple method like a PLS.
Okay. Just to rephrase this also, or to emphasize, there's one more question on the amount of volume per scan. I think you did answer this already. Most will attempt to set the measurement duration for a single scan or one spectrum to be equivalent to about one tablet volume. Is that right?
It makes absolute sense, yeah.
Perfect.
It's a common approach.
Okay. Just taking a brief look at the questions to be sure. Then, in your experience for the NIR and the bin blending, that has recorded quite different results when tested against HPLC samples from time to time. So, what is there the accuracy expected? Did touch this already, but do we have an answer on this? Owen, yes, go ahead.
Yeah. So, in the case of bin blending, let's kind of think about what we're measuring and what we're comparing to, and then let's take the case of the feed frame because I think that's a bit of a different situation. So during bin blending, we're making measurements in the process, and well, you may during, say, a validation run or some early technical batches, rotate the bin 10 times, 20 times, stop, thief sample. You'll have made a NIR measurement typically once every rotation there. Repeat, do this out to 100, 200 times, and then compare the variance of the HPLC measurements to the variance we see in the NIR method. In general, although I won't say always, in general, we know that thief sampling can have some biases. So I've seen people make 12 measurements, get unusual results in the HPLC. The NIR says everything's fine. They go back, they do some sort of resampling, or they take multiple samples, take the samples available for retest, and retest shows, okay, no, actually the NIR tended to be a bit more true. I think the situation here is a bit different for the NIR and the feed frame. We are no longer in a situation where we're mixing and we're able to pull tablets that can be useful. I think here it is uncommon, from my experience, for people to take a tablet, routinely measure it via chromatography during the manufacturing process as part of an IPC. That is less common than routinely taking thief sampling. You may take tablets at the end of a process for content uniformity, but as a blend uniformity IPC, that's a bit uncommon. In general, the NIR method can have biases. We tend to assume just a little bit of risk there. So early in development, I think there's probably more of a risk that the NIR method ends up being biased relative to the HPLC for these sorts of tablet measurements. We can do some sort of model update and hopefully true it, reprocess the data, and see if we can get a true result. It's a bit of a different situation. There are ways to handle it. But that's probably getting into getting into a topic or a discussion all onto itself.
Right. I think we can take one more question just because it has already been partially addressed. There was a question, can we simply monitor MBSD, so moving log standard deviation, in the feed frame as done in the blender? Is that not enough? I remember you did show a slide on explaining on the different methods applicable. Maybe also Adam wants to take the question coming more from the calmetrics side.
My presentation got a short correction in this topic, so that I would leave with Adam or with Owen, probably the more better one to answer.
Yeah, I can make a general comment here. So it really depends on what the goal of your feed frame implementation is. Generally, we are looking for the actual true quantitative value of the drug product, which moving block standard deviation won't give you. You could feasibly use it to demonstrate process control. But generally speaking, people are more inclined to want the actual quantitative result for CU confirmation. So you can certainly use the technique, but it's not common for feed frame implementations. Owen might have, I don't know, Owen, if you have any follow-up comments about that.
I would recommend people take a look at the paper I referenced on Pfizer's fusion approach, which does use a quantitative method, but I think it's more looking for deviations in the quantitative method and using soft sensor information to decide whether or not a process is on target. I do think that minimizes the risk of small amounts of model bias to new lots of excipients, new lots of API causing over or underestimation of blend potency, but will still give you the sensitivity to detect when a process is deviating from a state of control.
Thank you. Thank you for the two answers. We are at the end, in fact, already 3 minutes past the original scheduled time. With that, I think it's good to prepare for closing this webinar. I'd like to remind everyone that in 1 week, there is going to be a follow-up. So we did have a few questions unanswered. That is what the next week we are happy to address. Uwe, if you want, you can share that slide again with the details on the follow-up meeting. And with that, I'd say thank you, everyone, for your interest and your attention. Thank you to the presenters for the two very interesting presentations, plus the quite intense question and answer round. And I look forward to the next SentroConnect Live number 3 that will take place in 2026. Again, thank you, everyone. See you in the future. Bye.